So, what mechanisms can cause a change in a gene pool? What can actually change those genotype frequencies? What can change those allele frequencies? As we saw with the moths, those brown moths became more successful over time than the white ones. What causes that change? And there are a couple different things that can cause a change in a gene pool. There are three main causes that I'm going to teach you. Number one is genetic drift. You've kind of heard me throw that term out there. I'm going to actually teach you what genetic drift is. Founder effect is coming into play in this category. The bottleneck effect. Love the bottleneck effect. It's super cool. I'll teach you that. Gene flow is a second category that can cause evolutionary change. Students get gene flow and genetic drift confused with each other. I'll teach you the difference. And number three, the beautiful natural selection example that I have taught you. And those four criteria that must be met for selection pressure to account for a certain trait to make an individual more evolutionary fit, hence pass on their traits. They're more likely to pass on their traits to their offspring over time. Beautiful natural selection. But that doesn't mean that natural selection is the only cause of evolutionary change. Appreciate that I'm going to teach you gene flow right now. I'm going to teach you genetic drift right now. These are non-natural selection examples that can cause evolution to occur. Natural selection is the most important to consider because it is the only process that promotes adaptation. Adaptation is when individuals, as I've accounted for already with the moth example, becoming more evolutionary fit in their environment. The other two, they don't really lead to an individual becoming more evolutionary fit and more successful in their environment. Let's discuss why. So the first cause I want to discuss with you that leads to evolutionary change is genetic drift. Genetic drift is basically chance. It is completely random that that individual is going to move on and be successful in making offspring, having reproductive success, to promote their alleles to go on across generations. It's just random. There's no other selection pressure going on there for those alleles to move on than just chance events. So let me go ahead and describe what I'm talking about here. Here, get my pointer. I have the same flower population that we covered. I have the red flowers, homozygous dominant. I have white flowers, homozygous recessive. I even have some heterozygotes here that are also going to be red with that dominant R allele for the red color. Okay, and here I actually have 5 plants that I've boxed, and because of chance, completely by chance and for no other reason, those were chosen to move on and leave offspring. So I can actually see their distribution here in this now generation 2 population. If you look at the frequencies of the alleles, I can actually see the dominant allele represented by the letter P, as I taught you guys using Hardy-Weinberg. That's a 70% distribution of the red dominant allele, 30% distribution. Well, if I look at generation 2, it's actually 50-50. If we look at the alleles, not the phenotypes, don't just look at the color, look at the alleles, the letters, and you're going to see a 50-50 chance that those alleles are spread in generation 2 because of chance. This is genetic drift. Well, here I boxed randomly 2 plants. They ended up being 2 homozygous dominant individuals. Well, now those guys in generation 3, if they're the only ones to reproduce and make offspring, I'm only going to show homozygous dominant. So I have purebreds actually in generation 3. It's 100% of the dominant allele here in this distribution, and none of the recessive allele that just got lost due to genetic drift chance. The only other thing I want to point out, so not necessarily genetic drift, just Hardy-Weinberg, we wouldn't see this huge drastic variation in generation changes. Remember, Hardy-Weinberg is assuming that there's no evolution occurring. There's no selection pressure. There's no genetic drift. There's no natural selection. There's no mutation. And so Hardy-Weinberg is a nice way to show us what to expect without evolution. Evolution is definitely occurring when genetic drift is happening. You're changing the allele frequency. All right, so some examples of genetic drift. What could be this chance event that leads to only certain individuals leaving offspring? What do I mean by chance? What do I mean by random? There's two different effects that I want to discuss. The first one is the founder effect. The founder effect is when a small group from a larger original population moves to a new location. Because of that isolation, they then become more homogeneous. We only see the alleles of that small group. Okay, so for instance, here, I'm showing you an original population. It looks like we have a wide distribution in this gene pool. We've got burgundy color, we've got orange, greens, purples, lots of different alleles found in that gene pool. Well, let's say these guys right here, these guys are saying, okay, we're going to go and move on and find our own spot to live and be. Those are just greens and oranges. This is called the founding population. Well, you only have the green and orange alleles to then contribute to subsequent generations. Their descendants will be very different from the original population. This is called the founder effect. An example I'm showing you here, because necessarily these individuals didn't move on purpose. So let's look at these flowers here. Here I have flowers. I have all different colors of flowers. We've got blue, red, yellows. Let's say a bird, by chance, for no other reason, picks up some flowers. Maybe it was a byproduct of that bird trying to catch some prey, and those flowers got caught. Maybe for some other reason, but those flowers, looks like that bird has yellow and blue flowers. Move them to another location from its original spot. Well, now that new location is only going to have those blue and yellow alleles in their population. That's called the founder effect. It is a type of genetic drift. The bottleneck effect is another type of genetic drift where we have basically a drastic reduction in the original population because of a natural disaster event. Think of earthquakes, think of tornadoes, floods, any natural disaster event that's going to randomly prevent individuals from successfully reproducing, and so it really decreases their size. Those alleles won't move on, but the ones who did survive will. This is called the bottleneck effect. It's called the bottleneck effect for the reason in this image that you see. So here we have an original population. I can see different colors. Those are my alleles. So I've got orange, greens, purples, and I can see their distribution here in this bottle. Well, a natural disaster event occurs, and think of the bottle shaking out, and randomly these different marbles will fall out. Well, I'm going to get randomly, for no other reason. You're not going in there and picking out a certain color. They're randomly going to fall out from this bottle. It looks like a lot of these purples are falling out. I'm probably going to get a green falling out. None of the oranges came. After the disaster, this is the surviving population. Randomly, it looks like the majority purple alleles survived, one green. These will then contribute to subsequent generations. Those alleles will move on. The orange ones did not survive that natural disaster. This is called the bottleneck effect. They are both types of genetic drift. Gene flow is the next cause of evolutionary change. Don't get genetic drift and gene flow confused with each other. They are completely different. Genetic drift occurs due to chance events, as we just discussed. Gene flow is going to be when the gene pool changes. So an evolutionary change occurs in that gene pool due to emigration and immigration. So we are talking with those two different events, kind of either moving into or out of populations. We're talking about movement of individuals. So here I can actually see I have a population of brown beetles up here, and I have a population of green beetles up here, and it looks like this guy wants to go ahead and move. It's going to relocate and transfer its brown allele into this population. That's called gene flow. This brown beetle is basically contributing its own alleles, those brown alleles, to a population, that green population. Gene flow is very different from genetic drift in that it increases the genetic diversity in a population. This one used to be just green, but now because of gene flow, because of movement, I now have a brown allele in this population. I have differences now, green and brown. It's more diverse. The next statement reduces genetic variation between populations. What I mean by that is before, I had a very different population over here, green beetles, and a very different population over here, brown beetles. Well, now I'm getting a little bit heterozygous with those alleles, with the brown mixing in with the green. And so then the variation decreases. I now have some similarity, whereas before it was drastically different.